You know, every single living thing, from the tiniest bacterium to the trillions of cells that make you, well, you, is defined by a boundary. But this isn't just a wall, it's an intelligent, active border that separates the chaos outside from the absolute miracle of life within. So let's dive into this incredible structure, the plasma membrane.
I mean, it's a pretty fundamental question, isn't it? And on the cellular level, the answer is this amazing barrier. We need to get one thing straight right from the start, though. This is not some passive wall. No way. It's a dynamic, thinking gatekeeper, and it's fundamental to literally everything a cell is and does. Before we get into any diagrams or models, I really want you to see the real thing. Take a look. This image from a kidney cell shows the plasma membrane in all its complex, folded glory. See, it's not a simple, smooth bubble. It's a bustling, active surface, constantly working, folding, and interacting with its environment. It's beautiful, really. Okay, so to make sense of all that complexity, scientists use this, the fluid mosaic model. Just think of it as a sea of oily lipids with all these different proteins just floating around in it like icebergs. And that word fluid, it is absolutely key. This fluidity isn't just a random feature. It's what lets the membrane be so dynamic, to repair itself and to let its protein workers move around to wherever they're needed. This model is going to be our guide for this whole explainer. Now, what's so brilliant about this is the design of that lipid sea. It's a bilayer of these things called phospholipids. Each molecule has a water-loving head and a water-fearing tail. It's kind of like a huge crowd of people holding umbrellas in the rain. The umbrellas, those are our hydrophilic heads. They face the water on both the outside and inside of the cell. Meanwhile, the people, our hydrophobic tails, stay totally dry, tucked away in the middle. It's this simple bit of chemistry that automatically creates a stable, self-healing barrier. So clever. So with that smart structure in place, let's look at its first and most famous job, controlling everything that comes in and everything that goes out. This membrane is, no joke, one of the most selective gatekeepers in the entire universe. And there are really only two ways to get across this border. The first is passive transport. You can think of this like floating downstream on a river. It doesn't cost the cell any energy at all. Molecules just naturally move from where they're crowded to where there's more space. Then you have active transport, which is the complete opposite. This is like paddling furiously upstream. It forces the cell to spend energy, this stuff called ATP, to pump things against their natural flow. This image does a great job of showing the different kinds of that free passive transport. You've got tiny things like oxygen that can just slip right through. That's simple diffusion. It's kind of like how the smell of coffee just spreads through a room. But for other molecules, the membrane provides special doors. Some are just open channels, like a tunnel you can go through. Others are carrier proteins that work like a revolving door, grabbing a molecule on one side and then spitting it out on the other. Now, when it comes to active transport, the sodium-potassium pump is the absolute superstar. It's constantly burning energy to pump sodium out and potassium in. But here's the real aha moment. Its most important life-saving job isn't just moving those ions around. It's preventing the cell from literally exploding. It acts like the cell's bouncer, constantly pumping out stuff to control water levels and keep the cell from getting too full and bursting. Okay, let's move on to its next major role, because the membrane isn't just a gate. It's also the cell's antenna, its central communication hub for getting messages from the rest of the body. So how does it listen? It uses these specialized proteins called receptors. Each receptor is like a very specific lock, and it's waiting for one and only one type of key, a specific hormone or signaling molecule. When that key shows up and binds, boom, the receptor changes shape. That triggers a whole cascade of events inside the cell. They are, for all intents and purposes, the official translators between the outside world and the world inside. Now, a huge family of these receptors are linked to things called G proteins, which are basically molecular on-off switches. Don't worry about memorizing this table, really. Just look at the variety here to get the concept. You've got GS proteins that are stimulatory, they hit the go button. You've got GI proteins that are inhibitory, they slam on the brakes. And then you get these hyper-specialized ones like GT, or transducin, which you find in your eye and is responsible for the actual miracle of turning a single photon of light into a nerve impulse you can see. Just incredible. Okay, so we've established the membrane is this fluid sea. But the cell itself isn't some shapeless, flimsy blob, right? So why not? Well, that brings us to its third critical job, acting as an anchor for the cell's internal skeleton. The fluid membrane is actually tethered to this huge internal protein network called the cytoskeleton.
Think of the membrane as the fabric of a tent, and the cytoskeleton as all the poles that give the tent its shape and stop it from collapsing. This connection gives the cell its unique shape and makes it incredibly resilient. It's the perfect blend of flexibility and strength. And what's really cool is how adaptable this system is. For instance, have a look here. On the left, in a red blood cell, you see how proteins like spectrin and ankyrin create this flexible internal net. This is what allows the cell to squish and squeeze through the tiniest capillaries without breaking. But then, on the right, you see a totally different cell that uses a more rigid structure built with different proteins like talin and vinculin to anchor it firmly for a completely different job. So, we've seen the membrane as a gatekeeper, a communicator, and an anchor. But what happens when this absolutely crucial border is compromised? Well, the consequences are immediate and, frankly, often devastating. First, let's think about that communication system. What happens if a receptor is just blocked, if it can't receive its intended signal? The result is paralysis. Snake venom is a terrifyingly perfect example of this. Its molecules are shaped to fit perfectly into the receptors on our muscle cells, the very same receptors that are waiting for a signal from your nerves. The venom gets there first, it blocks the signal, and the muscle, it just can't contract. Blocking a signal is one thing, but what if something could actually hijack the system and lock a switch permanently in the on position? That's the devastatingly effective strategy of the cholera bacteria. Its toxin actually modifies that stimulatory GS protein we talked about, jamming it so it can't ever turn off. This one tiny molecular switch, when it gets stuck, forces intestinal cells to pump out catastrophic amounts of water and electrolytes, which leads to the awful symptoms of the disease. And finally, what happens if that structural link, that anchor holding the membrane's tent fabric to the skeleton's poles, just snaps? Well, then you got genetic disorders like hereditary spherocytosis. Remember that flexible spectrin protein we saw in the red blood cell? When that protein is faulty, the anchor is weak. The red blood cells lose their unique concave shape, and they become fragile, spherical, and easily destroyed. The result is chronic anemia, all because of a single faulty connection at the cell's border. So let's bring it all together. This elegant, fluid membrane has 3 core jobs that are absolutely essential for life. It's the vigilant gatekeeper, the responsive communicator, and the stabilizing anchor. And all 3 of them have to work in perfect harmony every single second of every single day. Which really just leaves us with one final, truly fascinating thought. All this transport, this signaling, this structuring, all these tiny intricate protein machines are operating within a layer that is just 7.5 nanometers thick. It is an absolutely astounding feat of efficiency and elegance, a true marvel of biological engineering.